This is a podcast about new crops. You're going to love it. Join us on The Cutting Edge, a podcast in search of new crops for Wisconsin. Welcome, everybody, to another episode of The Cutting Edge, podcast in search of new crops for Wisconsin. I'm Jason Fischbach, the Emerging Crops Outreach Specialist for the University of Wisconsin Extension, and today we talk hazelnuts. In 2022, Sean Mellenbacher and Tom Molnar published a must-read paper documenting the 100-plus year history of efforts to develop hazelnut cultivars for the eastern U.S. Most hazelnuts in the world come from European hazelnut, Corylus avellana, but that species doesn't grow very well in the eastern U.S. because it's not very winter hardy, and it's generally not resistant to a fungal disease here in the eastern U.S. called eastern filbert blight. We do have a native species that grows well in our region called American hazelnut, Corylus americana, but the nuts are really small and the shells are too thick. So, starting in the early 20th century, nurserymen like J.F. Jones in Pennsylvania, Clarence Reed in Maryland, and George Slate in New York made crosses between American and European hazelnuts with the hopes of finding plants with the best of both species: excellent yields, good kernel quality, good winter hardiness, and resistance to eastern filbert blight. Later, Carl Weschke here in Wisconsin, Cecil Ferris in Michigan, and then Phil Rudder in Minnesota gave it a run. All of them made great strides, but the thing with hazelnut breeding and what makes it so hard is it takes forever. It's one thing to go out in the woods and clone the best plant you find, which is basically how the Europeans did it, but if you're making crosses, especially between different species like we've had to do in the eastern U.S., it takes decades to come up with a proven variety, and these early guys simply ran out of time. To pull it off, we have to stand on each other's shoulders, and today we get to talk with two breeders doing just that. They are next in line to carry the torch, so to speak. Emily Delorean with the University of Minnesota and Scott Brainard with the University of Wisconsin join me for an update on hazelnut breeding here in the upper Midwest. First, because I think plant breeding in general is so important to solving our agricultural problems, I asked them to explain how they each got into plant breeding. And maybe, just maybe, a high schooler out there listening will get excited and pursue plant breeding as a career. Then, Emily and Scott catch us up on the genomics work they are doing to speed up the plant breeding process. Enjoy. Emily, let's start with you. Do you want to introduce yourself and tell us your bio and how you got into plant breeding and what your position is now with University of Minnesota?
So when I was a little kid, I didn't even know what plant breeding was. It wasn't like I decided one day that I was going to be a plant breeder. I kind of accidentally found this path. And when I first started college, I knew that I wanted to do something to help society. And initially, I thought that the best path would be for housing. And I was actually an architecture major for the first year. But I soon realized that I wouldn't be able to do as much good in that field as I wanted to. And I was kind of casting around for something else, and I stumbled on how powerful genetics are in that realm accidentally. And that was actually through Australian shepherds. Do you know what Australian shepherds are? The
No,
dog that kind of
yeah, right. Okay.
so there I was reading this web page that they Australian shepherds are famous for this color pattern called merle, and it's sort of the speckling, where they are the silver and they have kind of black speckles over the top of it. And all merle Australian shepherds are heterozygous. They can only have one merle gene, and then the other gene has to be the functional wild type version. And if you breed two merles together, what ends up happening is you get solid white puppies that they have deficiency in pigment even in their ears and eyes, and they are deaf and blind.
Wow.
And as an 18-year-old, this just blew me away that something that could be so innocuous as just a color pattern on the dog can cause such severe issues if not handled well. And it was really that moment that I wanted to study to know more about genetics and see how can genetics be used to do good and also to limit the bad that it can do. And I found at Colorado State University the plant breeding and genetics major, and I just kind of dove in headfirst into it and quickly started working for the bean breeder there, Dr. Mark Brick, and was really blown away with beans in particular because Most beans that we encounter every day are really just two kinds, are the pinto beans and the black beans. But there's an incredible diversity of beans grown in the rest of the world that we don't really have here. And so that was kind of the initial interest. And also, there were so many different species of beans that we don't eat here. And there's been multiple domestication events. And Mark was a wonderful mentor and encouraged me to keep going with this. And he was the one that encouraged me to go to grad school and really become a plant breeder. And from there, I went on to do a master's with Jan Leach at Colorado State University. And I was looking at rice genetics and particularly a pathogen to rice called bacterial blight, and looking at the gene to gene interactions between how the pathogen overcomes the resistance in rice, and then the rice develops new resistance, or we find new resistance to deploy to keep this our rice healthy and able to grow in these areas with immense disease pressure. Um, yeah, and from there went to a PhD with Jesse Poland at Kansas State University, where I was working on international wheat breeding with the International Maize and Wheat Improvement Center. It's called CEMIT in Mexico. And we were using genomic selection models on a very large scale. We were making predictions for thousands of lines every year for traits that are really difficult to phenotype, such as end-use quality, so the bread making ability of wheat. Um, and really just unlocking the power there of using those genomic selection models, which we will be using in hazelnuts, which Scott's been working on. Um, and a side project that I had in there was working on wheat wild relatives, the ancestors of wheat. And we were the Wheat Genetics Resource Center at Kansas State University is crossing the ancestors of wheat to domesticated wheat to bring in new genetic diversity, especially for climate resilience. And I was looking at the induced quality traits for that and just the unbelievable diversity that you have in the wild ancestors. And that is kind of what's fueled going into the hazelnuts, working with the American hazelnut. Yeah, so now I'm here at the University of Minnesota. I'm working with Lois Brown, and we are trying to apply some of these concepts to hazelnuts to domesticating a new species of hazelnuts and really using hybridization for that and applying genomic selection models because the timescales on hazelnuts, with these being a long-lived perennial, we're working with immense timescales, and genomic selection really helps us speed that up quite a bit. And developing other tools to hopefully have hazelnuts widely available in the upper Midwest.
My foray into plant breeding is hazelnuts, and I think the first thing that you learn is, wow, this takes forever
Yeah.
to do any of this. And I would imagine the same is true even for annual crops, right? But it does seem like, given that time scale, that early plant breeders, rapid genetic gain with the main staple crops, corn, soybeans, wheat, and that was where all the resources went. That's where the training programs were. That's where you did your postdoc, right? And it feels like the sustainable agriculture track, which kind of has been developing in parallel to that commodity crop track, they're starting to intersect more, where younger plant breeders and maybe even funding programs are really thinking more about diversifying away from just simplified commodity crop investments. In other words, more than just, and that could be, like you said, looking at um, older lineages of plants to bring more diversity back into those commodity programs, but then also looking at whole new species like American hazelnut or whatnot. Do I have that right, or is it still business as usual?
No, I think you're absolutely on the right track with that. And that was something, even as a student, I was picking up on it, though I didn't really understand all the forces behind it, because I did look at other crops to study besides wheat. I was interested in especially healthy crops like nuts and beans, but all of the resources, so much of the funding was going towards wheat and corn, which I looked at it as like I could go learn all of these cutting edge techniques in these crops where there's lots of money for it, and then I can come back and apply those techniques towards these other crops that need them. I think the commodity crops world is realizing also that part of breeding for climate resiliency is bringing in all of the genetic diversity and going back to the ancestors, uh, because the bottleneck that we've experienced with by making these rapid genetic gains has kind of limited us in some ways.
Yeah. Put us down a path that maybe we have to backtrack a little bit on, find a new
Yeah,
path.
exactly.
Scott, you want to give your background? You're currently in Virginia. Do I have that right?
That is correct. Yeah. My name's Scott Brainard. So I'm living in Virginia, but lived for a long time in Madison, Wisconsin, and that's where my professional home still is, working for the University of Wisconsin-Madison and nonprofit Savannah Institute. um, and yeah, I really enjoyed listening to that introduction from Emily and also your comment, Jason, about uh, the sort of parallel development of a sustainable agriculture track within agricultural research more broadly. That really maps on well to how I got into plant breeding. My like Emily, I don't come from a plant breeding background or even an agricultural background in terms of how I grew up. But when I was in college, I started working on organic vegetable farms, really just as a summer job. And I got really, I really fell in love with the work of agriculture, but also with the sustainable agriculture movement, and in particular at that time, I was really invested in organic agricultural practices, with sort of this realization or belief that there are problems with the way that conventional agriculture is practiced, the way most food, certainly in this country, is grown, is it has a lot of unintended consequences, and there's a lot of room for improvement, let's say. And a big motivator for me from the beginning was to see, what role I could play in trying to improve those systems, either by making them more sustainable or reduce their ecological impact more broadly, or improve human health and nutrition. There's lots of things we could talk about. But yeah, so after many years spending my 20s on veggie farms, I realized that I wasn't a very good farmer, and so I went to school for yeah, I went to a sort of general agronomy agricultural program in the Netherlands at Wageningen University. I like to refer to it as kind of a liberal arts degree in agriculture. I didn't have a specific thing that I thought I really wanted to study in terms of agricultural science, but I really wanted to get exposed, I think, to all of the different aspects of that discipline. And while I was there, I was really fortunate to take a class in plant breeding and plant genetics, just one class, and realized, this is fascinating, and something that just really felt like was kind of tying into a lot of my pre-existing interests. And so when I moved back to the US, I applied to PhD programs, and that's when I moved to the University of Wisconsin-Madison. I joined Erwin Goldman's root vegetable breeding program. It's a carrot, onion, and beet. While I was there, I learned for the first time also about agroforestry, and I obviously knew about the existence of tree-based agriculture, but it was while I was in my PhD program that I kind of got exposed to this as a form of sustainable agriculture, and so that led me to do a postdoc at University of Wisconsin-Madison in Julie Dawson's lab on hazelnuts, where I still work, working both on hazelnuts and on chestnuts. And that work has been led to and continues to really benefit from collaborations with the nonprofit Savannah Institute that is growing out a lot of the progeny families for some of the crosses that we've made. I'm sure we'll talk about that. But yeah, that's where I have been for years now, is doing this work on hazelnuts and more recently on chestnuts. And yeah, it's where I hope to spend spend the rest of my career, is maybe doing a few cycles of selection in these long-lived perennial crops.
Right on. Standing on shoulders for sure. So, listeners, let's do some history here. Let's circle back to 2007, 2008, when I would say the public sector hazelnut breeding programs in the Upper Midwest started. This was the launch of the Upper Midwest Hazelnut Development Initiative. At the time, it was just an idea and zero staff, zero money. And myself and Lois Braun, both trained agroecologists, got involved on either side of the border, myself in Wisconsin, Lois in Minnesota. And we quickly realized, wow, based on what the growers were telling us, the plants that they were growing just weren't good enough. There were individual plants in their populations that they thought were really exciting, but overall, the average yields and performance of their seedling plantings weren't good enough. Okay, so what do two agroecologists do with no real background in plant breeding? Start a plant breeding program. And we did it the old-fashioned way starting out, and that was basically try to assemble a population of plants, go out there and measure stuff, and hopefully find 1 or 2 or 10 of the best plants and copy those, and then evaluate those in multiple locations. And hopefully out of that comes a few that could support commercial production. And we think we have some, what we call the UMHDI first gen selections. And, as you probably heard us talk about many times, we're trying to get them propagated and out to growers. Okay. So that was a brute force project, more or less, more than anything. And we also quickly realized, wow, this is super inefficient. This is super expensive. This is going to take forever. We need better tools, and faster processes and smarter people, quite frankly, to move this thing faster, because it takes forever to breed any crop, but hazelnuts in particular, just because the cycles are so long. So we couldn't sit around and do it the old-fashioned way. Okay, so we assembled 3 populations, if you will, that became the base populations to use for some new tool development that we're going to talk about today. There's a population in Barneveld, Wisconsin, which is primarily American hazelnuts, though we'll circle back to that. We also have had a population in Stoughton, which was a putative full sibling family from 2 known parents. And then we had a really diverse population that Lois had created in Minnesota, which were hybrids between some of the selections from the Americana populations and the early hybrids with European Avalana selections. Okay, so now let's jump forward to 2022. That's when we last talked to Scott. And at the time, now Scott, this is the part where you interrupt me because I'm going to try to give an overview of what you talked about. So if I've got it wrong, jump in. At the time we recorded that podcast in July of 2022, you had finished the genotyping of plants from those 3 populations. And I believe you had what called short reads, 150 base pair chunks of DNA that you had sequenced from all those different plants. You were able to align those with the base genomes of, I believe, both Winkler and Rush, or was it just one or the other?
We did align them to both, but going forward now, we have decided to just use Winkler.
Winkler, okay. And then you were able to look for variants and found a bunch of SNPs, single nucleotide polymorphisms, which essentially become the genetic fingerprint for each of those plants. The SNP profile, if you will. Is that correct?
Yeah, that is correct, and that I think is a great. If none of that made sense to to folks listening, I think going back to that 2022 episode, you're reminding me that we really got into the nitty-gritty of, the chemistry of how sequencing
Yeah.
works these days, next generation sequencing, how you identify what are called molecular markers, so variations in the genome from plant to plant, why we do that in the first place, and, kind of some of the bioinformatic work that goes into identifying that. So that was a great summary. And if anyone wants to know more, we already did a whole podcast
Yeah.
episode almost about that. So
And
yeah.
Scott, I went back and listened to it. I was just blown away at what a good job you did bringing that complicated topic to a lay audience. So I definitely echo what you said. Listeners should go back and listen to that one. And it's a great crash course in some of this genomics work.
Yeah,
Um,
maybe
so
if I could just say,
yeah,
you were saying just a second ago that you and Lois did this the old-fashioned way when you first got started, and I was actually kind of struck at how well you described how, I mean, maybe Emily, you'd have some a different take here, but that what you just described is really the breeding process. And it's, it is the old-fashioned way because it's the way that people have been doing it for a long time, but it's also the way that um, breeding still happens in most crops is you build a diverse population, you evaluate it for the traits you're interested in, you use those traits to, pick winners and make new crosses. And a lot of the tools that we're developing, that Emily and I are both working on, are ways to make that process more efficient. But fundamentally, that process remains the backbone of a of a of many breeding programs that aren't doing just pure transgenic work, which we're not. And so I just want to kind of make that distinction that we are still doing what you might call traditional breeding. We're just trying to use advances that have been made in genotyping to give us a new layer of information, and also to make our phenotyping approaches more efficient as well. But yeah, anyway, I
yeah.
just wanted to make that
All
point.
right. So with that in hand, the plan was, as of 2022, you were going to take those SNP profiles, for lack of a better term, and develop the tools to make these more efficient breeding programs, right? So let's just review those tools. So one thing you could do with those SNP profiles was compare them across all the different plants that you genotyped. So you could take the Barneveld population and compare all of those different SNP profiles, the genetic fingerprints of all 600 plants or so that you genotyped, and you could understand how they're related to each other, right?
Hmm.
Same thing with the Stoughton planting, same thing with the Minnesota planting. So
Yeah.
that's useful to know. The other thing you could do with those tools is, let's say any one of those SNPs, if it's linked to or associated to a specific trait. So I think in the 2022 podcast, you used the example of large nut size, that all of the plants that had a large nut size also had the same nucleotide at SNP 522 or whatever, right? So you're able to maybe hone in on which part of the genome was controlling a particular trait. Do I have that right?
Yes.
Okay.
Yes.
So that's some more
Correct.
association analysis. And then maybe the third tool that you had talked about, and this was actually the one you were most excited about, was you could use this to be able to understand how easy it might be to improve any given trait.
Mm. Yes.
Okay, so we've got the association analysis. Are there have you sort of linkage mapping, if you will, relatedness, and then understanding how easy or the heritability or how easy it would be to breed for a trait. So let's let's hear it. What have
Yeah.
those tools, and maybe take one population at a time and maybe talk a
Sure.
little bit about that population and what you've learned about it, and then what you might be able to do with that population going forward. So maybe let's start with maybe the easiest one. Let's start with the Stoughton planting. And
Yeah.
so just a little background, we planted in 2011 at 3 locations in Wisconsin, basically the same planting. We got the seedlings or offspring from a breeder forest agriculture enterprises here in Wisconsin that were plants from a controlled cross between two of their top plants. And interspersed in that mix, we also planted American hazelnuts from the DNR. So that's what we planted. So, but we weren't totally sure what the genetic makeup was of the C. americana. And because pollen moves around so easily, we weren't 100% sure that all those offspring were from that particular cross. So what did you learn?
Yeah, so this, I would say the main conclusion or the main takeaway from the Stoughton planting is definitely in the bucket of what you were saying about learning or using genotyping methods to learn about the relatedness between individuals. But at Stoughton, what we were looking at specifically was to try to reconstruct pedigrees. Because, like you were saying, the plants that were planted there were seed that was collected off of clonal rows of hazelnuts that had been planted next to each other, with the goal of having the progeny being controlled cross, essentially. But the pollen was not moved by hand from one flower to another, which is, how, for instance, it's done in a lot of the creation of the families in Minnesota, where Lois had received pollen in a vial, from collaborators around the country and literally made the cross. With the Stoughton plants, there were these grafted rows of hazelnuts, and because hazelnuts are self-incompatible, the theory is that those plants should exclusively pollinate each other. But of course, there's this chance that pollen might come in from outside, and you get sort of a stray, errant pollination from some unknown plant. And what was really cool looking at the data from Stoughton is we were able to see exactly this pattern where because we had the genetic sequences of the parents, and we had obviously the sequences of all the plants at Stoughton, all these putative progeny. And we were able to find three categories of plants: the wild DNR plants that you mentioned as the sort of controls, the just unknown open pollinated plants from wherever. Then you had the full sibs, which basically they had some deviation from expectation that could be attributable entirely to genotyping error, like less than 1%. And then you had some plants in the middle, and those turned out to be half siblings. So all of the seed was harvested off of these grafted rows. So of course there was, at least one of the two parents always represented, but we were able to quickly pick out the plants that were results of this errant pollination event, like I was saying. And those would have, a certain amount of error or deviation from expectation, but not as much as the wild Americana. So that was really cool, just to sort of test out this genotyping method and see that it is able to confirm what you were expecting essentially to happen, and also figure out the places where things maybe didn't go as expected.
Okay, so let's move to the Barneveld planting. And this one to me was really interesting because what it is right now you can, as a landowner, you can go to the DNR, Wisconsin DNR, Minnesota DNR, and you can buy American hazelnut plants. The way the DNR grows these out is they have people, just regular people that go out into the woods and harvest seed, bring the seed to the DNR grows it out. So they don't really know anything about it, right? So is it coming from this, if you're going to go out in the wild and collect hazelnut seeds, you probably year after year go to the same bushes because they tend to be the highest yielding, right? So maybe you have a less genetic diversity than you think, but it's also wind pollinated. So dad is coming from who knows where in a planting, right? So we really didn't know how diverse this particular planting was in Barneveld. And then the other part is uh, we well, and maybe I'm getting the cart before the horse, but how related Americana is to Avellana, if you will. There's a concern that if we bring in hybrids or Avellana into the Midwest, that we end up contaminating in air quotes, the Americana population, but how much Avellana is already in Americana. I mean, they're
Mm.
different species of
Hmm.
the same genus, so how different are they? And if they're not all that different anyway, how big of a concern is it that we're using Avellana or breeding, right? So there's all those big questions about Americana that I think were really interesting going into this. So what have you learned so far?
Yeah, well, yeah, I agree. I think the Barneveld planting is really fascinating, because it is, hundreds of DNR sourced putative Americana plants all in one spot, not too far from Madison. Um, and so, like you said, it's not necessarily, or it almost certainly is not representative of total Corylus americana genetic diversity, across the range. There have been other collections that exist. Jason, you've been involved in collecting in some of those collections yourself. Mike Demchek, Tom Molnar at Rutgers has like a sort of intentionally geographically diverse collections of Corylus americana meant to sample across the whole range. Um, this is more of a snapshot of just what's coming through the DNR, but it is at least theoretically wild americana. So there's two interesting conclusions I think that we were able to draw from Barneveld. One was we took the data from Barneveld, the sequence data, and we put it on the same plot, if you will, with sequence data for a lot of the what are called the Upper Midwest Hazelnut Development Initiative hybrid varieties. So this is kind of what you were describing as the Gen 1 plants that came out of this initial almost like mass selection program that you and Lois did, Jason, which has identified some really promising plants. So we sequenced those as well. We sequenced also a bunch of plants from Minnesota, which we'll talk about in a second, and also the parents of the families of the plants at Minnesota, which included a lot of pure Corylus avellana. Again, we'll get to Minnesota in a second, but those full sib families had paternal parents, so pollen parents from Oregon and Rutgers, pure European hazelnut parentage coming in. Basically, the Barneveld plants all cluster very closely together in one corner of this plot, and let's call that the American hazelnut corner. A lot of the UMHDI varieties, the kind of winners from this mass selection program, are also right over there with the Barneveld plants. They look genetically and, of course, we should say phenotypically, a lot like American hazelnut. On the other side, you've got, on the far right-hand side, you've got the European hazelnuts varieties, the few European varieties that we ended up sequencing, the parents of the Minnesota plantings. And then in the middle, just like you would expect, you see these F1s, these full sib families that are growing at Minnesota, because they are crosses between American hazelnut and European hazelnut. What's really interesting, I think, is that there are a couple of the UMHDI varieties that are actually right in line with the F1s. So Rose 9-2 is an example of this. It's one that sort of genetically looks like a real hybrid. We don't know the full pedigree of Rose 9-2. We don't have good records for exactly what plants were crossed together to make a lot of these UMHDI varieties. So we can't do the sort of like pedigree checking that we did at Stoughton because we don't know which ones to even include in the analysis. This is something we could do down the line if we sequenced a whole lot more plants. But so we don't know for sure, but there's certainly quite a bit of variety, or let's say genetic diversity, in the UMHDI plants called, putative or proposed varieties that are coming out of the breeding program. Some that look genetically very American, some that look more hybrid esque, and I think that's really cool. I think that is not a necessarily like end of the this isn't like a paternity test that would hold up in court, but it definitely shows that there is quite a bit of genetic diversity in the UMHDI breeding program. The last thing I'll say is that at Barneveld, if you just zoom in on Barneveld, because you can't really say much about the genetic diversity in Barneveld when you stack it up against Corylus avellana. It's good.
Yeah.
Like the difference between those two is much greater than the difference within Barneveld. But if you just look at Barneveld, a second ago I was talking about this sort of like 0 to 2 scale. Zeros in two plants are totally unrelated. Two, they're kind of like the same plant. The plants at Barneveld are very unrelated to each other. They are, like we were saying, the product of a sort of random seed sourcing operation that is put on by the Department of Natural Resources. So we don't know exactly who gave these seeds. Probably they came from specific geographies. So it's more telling us about the sort of relatedness or the degree of relatedness, even within specific regions of the American hazelnut native range. There is a lot of unrelatedness between any given two hazelnut plants that you might come across in the wild. That would be my provisional conclusion from that, which makes sense because, again, this is an obligate outcrosser. It has a very rigid self-incompatibility mechanism that also excludes even crossing with, semi-related hazelnut individuals. This whole S allele system doesn't just prevent self-pollination, but it prevents crossing with closely related partners.
Which is consistent with what Mike Demchek found, that there was more diversity within a given location than there was across locations, right?
Yeah, I can't really, I don't think that this conclusion supports maybe, let's say, the strong version of that
Yeah.
statement, that the within is greater than the between, but it definitely seems to be the case that the within is at least as great as the between.
Got it.
It's there is just, even within narrow geographies, a tremendous amount of genetic diversity. And yeah, I
Okay.
think that's also very interesting as well.
All right. So we've got Americana. It's pretty diverse, but is it diverse enough for the traits that we're interested in to be able to make genetic improvement without introducing Avellana? And maybe that brings
Hmm.
us to, have you learned anything from the association analysis approach with the Barneveld population? Like,
Yeah.
can we improve, and this gets to your question too, how easy will it be to improve these traits? Can we improve nut size in American hazelnut beyond its average of like 0.4 grams,
Right.
using just American hazelnuts from what you've seen so far?
Yeah. 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 Kind of everything I've talked about up till now is, academic, intellectual curiosities. What about actually improving hazelnuts? What have we learned? So we did measure for all of the plants at Barneveld and Minnesota for multiple years a whole suite of nut morphology traits. We used a digital imaging-based approach where we would take pictures of a subsample of the nuts and the kernels, and it took forever and gave us a lot of interesting data about the size and shape of the nuts and the kernels, the percentage of the nut that is kernel, and yeah, so on a purely phenotypic level, there is quite a bit of there is substantial variation in Barneveld. I don't want to make sweeping claims again about what that means about the full extent of variation in Americana in general, but there are plants at Barneveld with zero point 0.6 sorry, let me talk about percent kernel because that's a trait that I know the numbers off the top of my head, and I think is really important. Um, it, Oregon State, which has a very well-established, successful hazelnut breeding program, has, I think it's 48% kernel is maybe their like sort of threshold for what they need to hit for a new variety. We have plants at Barneveld with 50 approaching 60% kernel. Not
Wow.
many, but there are some there. And they, of course, have smaller nuts than Avellana, but a lot of the nut is kernel. When you look at the overlap in the distribution for most of these nut traits with Barneveld on the one hand and these full Sib families at Minnesota, which again are 50% Avellana, the distributions overlap. Of course, the plants with Avellana in them have larger nuts, have larger kernels on average, but there is substantial overlap in those two distributions. So yes, there is quite a bit of phenotypic diversity at Barneveld. To the question of association analysis, and so association analysis, linkage mapping are two different approaches to trying to find, particular markers that are linked to a region of the genome that controls a trait of interest. We were relatively, I would say, well, we did the analysis. We found some markers that are linked to what are called quantitative trait loci, which is kind of a fancy word. It means a region that contains genes, but it's too large to say just gene, right? It might actually span multiple tens, hundreds of genes might be in this locus that we're going to call a QTL. We found markers that are linked to QTL that control these traits. But the effect size was not huge. And that makes sense because these are not, we went into this whole experiment sort of thinking that these nut morphological traits are probably under relatively limited environmental influence. Contrast that with something like bush height, which is going to be like really determined by the management practice, the soil type, the level of fertility, et cetera, et cetera. You can juice your plant and it'll grow really tall. The actual control over nut size and shape, based on previous research, we thought would be under relatively limited environmental control, which is why we picked these traits for an experiment where we were working with existing populations that didn't have, very robust experimental design. But we also know that these traits are quantitative. They're not like, there aren't like two kinds of nuts, big nuts and small nuts, or, kernels. Either your kernel is 0.4 grams or 0.6 grams. No, it's a quantitative trait, huge amount of variation, all within the range. So it's under the control, we would expect, of many, many genes. And every individual gene is going to have a relatively small influence over the ultimate performance of the plant. And that's what we found. We found lots of QTL for most of these traits, with each individual QTL contributing not very much to the overall variation in the plant. What we additionally did was a word that I think Emily brought up at the beginning and you were just mentioning is what's called genomic selection or genomic prediction. Instead of trying to associate a specific marker and trying to test whether or not it is physically, genetically linked to a QTL, you say, let's just start from the hypothesis that related plants are going to have similar performance. And then use these markers to guess how related the two plants are. I was actually just kind of basically talking about this particular concept of relatedness as something that varies from 0 to 2. And we can give each pair of plants a number on the spectrum using their markers and say how related on this scale are these two plants. Ones that are related, let's just assume they're going to be closely similar in terms of their phenotypes. Unrelated will be more distinct. And then we build a linear model that just predicts the performance of, and when I say performance, I mean, let's just say like the percent kernel for any trait. You plug in whatever trait you want there. We predict the percent kernel based on the relatedness between a given plant and another plant. And so obviously to train this model or to fit this model, you need to have plants for which you have genotypes and phenotypes. And we do in Barneveld and Minnesota. We phenotyped and genotyped all the plants, but then we can sort of pretend that we didn't phenotype all the plants and we just have its genotype. So we can artificially mask or delete the phenotype and see what is our accuracy. We know what the phenotype is, but let's delete it from the data set and just use this model that we fit and see what the correlation is between the actual phenotype, if we assume we got it perfect when we measured it by hand, and the predicted phenotype. And there, I would say we were pretty successful. We get prediction accuracies over 0.5. That might not sound very good, the correlation coefficient over 0.5, but 0 is chance, right? And 0.1 and 1 would be perfect. You never get perfect predictions. 0.5 is okay given the constraints of the populations that we're working with. Relatively limited sample size. Again, no clonal replication in these fields to control for environmental variation. So we're literally just not even modeling environmental variation. We just assume that it doesn't exist, which we know it does. And then we, are using just a couple years worth of data. Given all of that, the fact that we're able to make correlations between predicted and actual values of greater than 0.5 for these nut traits, I would say is pretty good. It definitely justifies using genotypic data alongside phenotypic data in your breeding program. Even if you were, because let's just say you're starting with a seedling and you're trying to figure out which seedlings in the greenhouse I'm going to put out into the field. You don't have any phenotypic data. If you had genotypic data, at least you'd have some basis for stacking the deck. You could put out the plants that you predict, and on average you're going to be right to a certain degree in putting out the plants that'll have the best performance. And I should say these models are really good at eliminating the worst plants, and they're not perfect at perfectly sorting the entire list, right? Like getting the exact value correct. But if you're in the right ballpark, it makes this predictive technique very useful in terms of like culling the bottom 10 or 20% of your population or something like that.
Yeah. Good stuff.
Yeah.
And
So
and I
that's
had
what we,
something to add to that, Jason.
Yeah, Emily, go.
With your initial question of, and I think what you were getting at is why are we using Avalana when we have Americana, especially to get larger nut size? And I
Yeah.
think one of the things that we are really lucky to have is Avalana has been domesticated for thousands of years. And part of that domestication process was breeding for a much larger nut size. So like if you think about corn and its ancestor teosinte, teosinte was a very small kerneled corn with very few kernels, but domestication increased the number of kernels and the size of the kernels. And we get to use some of that. The same thing happened in hazelnuts, and we're able to use that from the European hazelnut. So, work that our ancestors did over thousands of years to increase the nut size for the European hazelnut, we're able to kind of skip those steps and on a much smaller timeline, bring in those genes into Americana. So now we can have all of the benefits of Americana, the cold tolerance, the Eastern filbert blight resistance, and bring in large nut size from the European hazelnuts. And
Perfect
maybe we can
segue.
also,
Yeah. Perfect,
okay.
perfect segue into the Minnesota population. Um, so a little bit of background. The population was started in 2014, roughly, when Lois took what she thought at the time were the best of the hybrids that had come from our selection process that started in 2007. And then in an effort to get larger nut size, she crossed those top hybrids with uh, European hazelnuts. And ideally, it was selected improved varieties of European hazelnuts, and then planted those at Rosemont, Minnesota, roughly 2,000 plants, I think, um, 2014, 2016. And then from there have been more plants produced, but we get to that. So Emily, that's basically all I know about the Rosemont planting. Can you tell us more about what's out there, what you've been doing with it, what you've learned from it? Scott, same thing. But Emily, you've been at it for a while now with that particular planting. So what have you learned, or what have you been working on?
Yeah, so the planting that you're talking about from 2014 to 2016, those were the top selections of the Upper Midwest lines crossed to primarily the Oregon State University top lines. So we had Jefferson and McDonald, who are really good performers in Oregon and have really large nut size, really good percent kernel. And when we crossed them to the American hazelnut, we did see an improvement. We have some that have really large nuts.
What about EFB? That's what's always scared the pants off of us, is that we know if we bring in European hazelnuts, particularly those that are susceptible to EFB, and cross with American hazelnut, that we're going to create offspring that are susceptible to EFB, right in the native range of EFB, where who knows how many isolates or strains of EFB we have, and if we deploy those out on growers' farms, we're just asking for major problems. So what have you seen in those populations with respect to EFB?
Yeah, so at the EFB, I know quite a bit more about that because that's something I'm directly looking at right now. Is with those crosses, it was thought that the maternal parent, the upper Midwest line, is resistant to EFB because these should be lines that have been already selected for EFB resistance. And the American hazelnut co-evolved with EFB, so it should have lots of resistance already naturally occurring. Whereas the European hazelnut, which did not co-evolve with EFB, because there's not EFB in Europe, where the European hazelnut's from, uh, and in Oregon, there wasn't EFB either, which is why that's where we were able to grow the European hazelnut. But there is predominantly released in the Oregon germplasm because there was one strain of EFB that was accidentally introduced in the 60s or 70s. They had found a single resistance gene that conferred resistance in European hazelnut to that strain of EFB. Whereas in the Midwest, we've got many strains of EFB, so they can overcome that resistance quickly. Uh, so the crosses, they're the Oregon European hazelnuts that we're using in our material are theoretically susceptible to all the different strains of EFB we have here, even though they carry a resistance gene to one single strain. So one of the things that we saw when we crossed our resistant hazelnuts to the technically susceptible hazelnuts is about 50% on average of those progeny were still resistant to EFB, completely resistant, no EFB at all on them after 10 whole years. But 50% did develop EFB, and many of them died from it. So we actually are able to maintain a high level of resistance even when we're crossing to susceptible material, uh, under the assumption that our resistance is quite strong coming from the Americana backgrounds.
With this particular Rosemont planting, which was really the public sector's first foray in the Midwest into crossing Americana with Avalana. We've got the top 30 that'll probably get winnowed down to the top 5 after we run these replicated yield trials, and those will be the second generation of improved plant material to come out of our breeding program. Um, are we done with that planting? Are you guys done with that planting? What else do you hope to learn from that planting going forward?
Yeah, so uh, we were initially going to start making culls, taking out the plants that we didn't want anymore that were not being advanced and that we were done with. But Lois and Mark and Les have done such an incredible job maintaining this planting for the last 10 years that I argued that we should keep these plants for another year because there's so many other phenotypes that we can get off of these plants to help develop these genomic selection models, especially with them being really mature, really well taken care of. Things like the plant architecture, canopy size, the EFB resistance, which is what I've personally been measuring. Um, and probably we won't be able to get much off of the yield because that is such a labor-intensive trait to measure. But some of these other things, because we are looking really closely at, or we will be looking really closely with genomic selection for things like bush size and architecture and EFB resistance, so that in future years, when we are genotyping all of our seedlings before they go out to be planted in our breeding nurseries, we can determine, okay, like these plants are going to be way too big for mechanical harvesting, so we should not plant these. Or these plants are probably going to be susceptible to EFB, so we shouldn't bother planting these either. And then we can really plant the material that we have hope will perform well and that we can make a really nice top selection out of in the future. So right now we're hanging on to the Rosemount plant so that we can get more data out of them, especially because with them being already 10 years old, they are really valuable for that.
Yeah, I guess, right? Because once you have the genotype, you only do that once and you've got it. So you might as well get as many different phenotypes or phenotype as many different attributes as you can to maximize the use of that genotype in prediction, right? It's essentially
Exactly.
the idea. Yeah. Okay.
Yeah,
Um, okay,
I
Scott.
totally agree with Emily on that. Yeah, I think there's been so much invested in those families, that, and this is kind of true of perennial crop breeding in general, is it takes so long to get a planting to the point where you can measure, where you can even start measuring certain traits. EFB takes a really long time to express itself. Resistance can be something that breaks down over time. Yield obviously changes markedly, or typically it goes up, over the first decade, but there can also be crossover events. We don't really know for any given individual plant whether a really high producer in year 4 is going to be your top yielding plant in year 14. So just like you're saying, Jason, if you already have the genotype, you've already got the planting so far, there's a lot of incentive to get as much trait data off of it as possible, because you can't do any of these. This is kind of what I was saying at the beginning about how this is really just improving the way we do traditional breeding. You can't do any of this if you don't have some plants, some populations that you have advanced to the point where you're able to get good phenotypic data off of them. So, yeah.
Okay, let's transition to the future and the new stuff that we've got. So that planting at Rosemont was in some ways, in terms of choosing parents, was just like a best guess, because at the time, even the hybrids from the UMHDI, we didn't know the top ones until 2017, 2018. So Lois was kind of guessing which were the best in choosing the hybrid parents. And to some extent also for choosing OSU, Oregon State University, pollen parents, right? And so we're obviously getting a lot out of that Rosemont planting, but I would say the fun stuff starts now, right? Because in 2019, 2020, Lois then started making, a couple things happened. We had an actual better handle on what were our top hybrids. So Rose 92, State Bend 76, ARB 71, Price W41. At the same time, Tom Molnar in the Rutgers program released some new Avalana varieties that had novel EFB resistance besides gas away, and were also selected in a little harsher environment. Now, the Atlantic seaboard is definitely warmer than here for sure, but it's definitely colder than Oregon. And so there was different Avalana parents that could be introduced in the breeding program. So let's made plant or crosses with that. And maybe even better is we actually had breeders working with us at the time rather than just 2 agroecologists making up as we go. And so designing the site layout, designing the crossing blocks, designing in terms of the planting of the progeny, which is the Valley Farm in Spring Green, in partnership with the Savannah Institute. So Scott, can you tell us about the Valley Farm? What's there? So there's new linkage mapping populations, and there's what's this thing that's called the DILL. And then Emily, when Scott's done, can you tell us what is at Becker now and St. Paul, similar populations, but and then what you're hoping to get out of those two? And I guess one specific question is, we planted all those seedlings before we had prediction models. So might we be able to, instead of phenotyping, ideally we wouldn't have planted some of that material in the first place. We could have thrown it out in the greenhouse first, right? Based on the prediction models. But can we just not phenotype some of those plants that are out there after we sequence those and use the prediction models to try to save phenotyping labor? So anyway, that's lots of questions. So Scott, let's start with you, the Valley Farm. Tell us in plain English what a DILL is, what you're trying to do with that population. Yeah, go. And then we'll have Emily talk about the Becker population.
Okay, yeah, I'm really excited about what's called the Valley Farm population. So this is, like you were saying, Jason, a collaboration with Savannah Institute. They were lucky enough to acquire a whole lot of farmland in the Spring Green area. Some really beautiful parcels that go up into the valleys. This is in the driftless area of Wisconsin, just about an hour west of Madison, but also down in the Wisconsin River Valley, hence the name, a large piece of land that is exceptionally flat, pretty uniform soil types. Because it's down in the valley, pretty rough, windy, cold, so good environment for selecting for cold hardy plants. But yeah, hazelnuts are cold hardy. So this was a good place in terms of the lack of topography, the overall available acreage that's there, the relative uniformity of the soil type, to do or to plant a long-term training population for doing genomic prediction, but also lots of other experiments. Like I was saying, when I started my postdoc with Julie Dawson in, don't even remember, 2020, we had lots of ambitious questions and I've been describing the results of that research. But of course, we had to work with the plantings that already existed. So we were Stoughton, Barneveld, these plantings in Minnesota had all been built for other reasons. Stoughton was a farm. Barneveld was really just a landowner's, sort of hobby project to do native plant restoration. He ended up planting rows of hazelnuts. And then, obviously the Minnesota planting is a breeding program, but was not set up for these kinds of experiments. So with Valley Farm, we were trying to set things up deliberately to, a decade from now, be able to answer some of these questions even better, with more precision, and use these methods even more accurately. So the big differences at Valley Farm compared to these other plantings are, one, the plant spacing is much greater. So there's 20 feet between the rows and either 10 or 15 feet between the individual plants. So this is not how you would grow hazelnuts in the upper Midwest if you were doing hedgerow production, but it means that the plants can get quite large and not compete with each other. They won't be shading each other out. You'll be able to see the plants distinctly. It won't be unclear which plant is which. They won't be competing for nutrients, for a long time. And we're managing the site very deliberately to, kind of maintain a uniformity across the field, but also, not stress the plants in any way. We're not trying to do, we're not trying to test for any sort of stress resistance. We're trying to grow uniform, healthy hazelnuts from the beginning. The other big thing we changed is that we planted randomized clonal checks all throughout the field. So we're very fortunate to, again, be sort of standing on the shoulders of Jason and Lois, giants in their own right, and we were able to get layers of all of these really cool upper Midwest varieties. So we have, instead of just a field of seedlings that are each genetically unique, we have about 12% of the overall planting are some layer or another, some clone or another. And that's really great because it allows us to model the environmental variation that does invariably exist in the field. And and then the last thing that kind of distinguishes this is what you were mentioning, Jason, is the intentional selection of parents. So we were really leaning on Lois and you, Jason's expertise in all of this and picking the parents themselves. But what we did in big picture terms is split the field into two pieces. On one side, we crossed Midwest parents by Midwest parents in this structure called a diallel, where we picked 6 parents and we made every pairwise cross between all 6 of them. So that's 6 times 5 divided by 2 total number of families. And that is a really common technique in plant breeding to try to, you can do what's called multi-parent QTL mapping. So very similar to the QTL mapping I was describing earlier, but now you've got more parental haplotypes floating around. So you can do more interesting stuff there. You can also use this mating design. This is a kind of mating design, and it allows you to figure out which parents are sort of generally really good. That's called general combining ability. So which parent, no matter who it's crossed with, results in really good progeny. And then specific combining ability. You can say this particular cross, for whatever reason, is generating incredible progeny. And, maybe we want to run with that. Maybe we want to explore those full SIB families a little bit more. On the other side of the field, we have some very large full SIB families that are crossed between upper Midwest parents and pollen from the Rutgers breeding program. The Rutgers breeding program, again, is using mostly C. avellana in its pedigrees. They are doing some hybrid breeding, but they have a lot more diversity of EFB out there than Oregon does. So EFB resistance has been a #1 breeding target for Professor Molnar. And also, it's not Mediterranean, at least, it's not as Mediterranean as the Willamette. So they are breeding for a little bit more cold hardiness, resiliency, or they are able to do that a little bit more. And so those are some cool families just to have more diversity in the program. And yeah, that is what it is. It's also large. It's about 15 acres of hazelnuts at this wide spacing. It's about 1,700 total plants, which is much larger than Barneveld, much larger than the individual 3 families at Minnesota that I was looking at. So that's also really great in terms of just getting the total sample size up. And then over the years, collect lots and lots of trait data on this planting. It's set up for the long haul so that, we'll be able to get per plant yield, hopefully, off of these bushes for many, many years and be able to see, how does early year yield correlate with mature yield? If we can figure out how to estimate biomass production every year using drones, we can look at cool things like yield partitioning, see how plants that put more energy into woody biomass, does that affect yield? So we'll be able to get a lot of hopefully very informative trait data over the coming years. This planting is just beginning to start producing. A few hundred plants produced a crop this last year, and yeah, so it's
So is every
exciting.
plant in there part of a tool development process? In other words, once you have the genotypes, will you predict what they will be, maybe validate them with 1 year of nut size data and say, too small, that plant's done?
Right. Yeah, so there's a few things that we will do. We will make selections from this planting. For sure, out of 1,700 plants coming from controlled pollinations, there are going to be some really cool plants that we are going to want to clone and, put into multi-environment trials. And this will be the next, this is looking far into the future, but the next set of varieties that we want to release to farmers, some of those might come from this planting. The other thing that we will definitely do is combine the genotypic data so we are going to phenotype this entire planting. It's set up for long-term phenotyping, but we will also genotype it. And so when it comes time to make crosses off of this planting, we will combine the genotypic and phenotypic data together to make even better predictions. So this is something I didn't really mention, but the phenotypic data is not perfect. The phenotypic data is a product of the true potential of the plant and where it happens to be growing, the little microenvironment that it experienced that year. Maybe somebody ran over it with a tractor and that, maybe it's planted on top of a decomposing animal. It's got a lot of extra fertility.
Blue jays stole half the nuts and we didn't know it when we harvested.
Yeah, right. Pest outbreaks occur. They are not uniform. So the phenotypic data is great, but it's not like, some sort of like truth. And so if you add the genotypic data to the phenotypic data, you can make even better predictions using the two together than any one alone. So we will do that. The other great thing about using genotypic data when picking parents to make future crosses is you can balance the desire to produce, you can make a prediction about, okay, so there's 2 things you want to do in a breeding program. You want to make progress. You want to create better plants for whatever that means, whatever trait you're interested in. You also want to preserve a certain amount of diversity in your breeding program. You don't want to end up with a whole bunch of inbred plants that are all perfectly related to each other, or you won't be able to keep making progress. And hazelnuts are not fans of inbreeding. They have a lot of inbreeding depression. So with the genotypic data, you can do 2 very cool things. You can predict not just with more accuracy, the like particular performance of a plant, but also what the average value of its progeny family will be. That's typically what's called its breeding value. You can also figure out how related these plants are to each other. And you can kind of build a balancing test where you set certain thresholds for how much diversity you want to maintain in your breeding program and what, sort of like minimum breeding values you want to achieve. And you can make crosses that will point you in that direction, where you can balance these two against each other. Once you've made those crosses, we will also be able to use this prediction model that we're building in order to, kind of do the very classic thing where you've got a whole bunch of seedlings in the greenhouse. You've only got X number of acres to plant out, X number or Y number of plants. So you can use the prediction sort of blindly in this way where you don't have phenotypic data to just figure out which ones are probably going to be the best. And so those are the three big things that I would say we hope to do.
Emily, can you tell us about the Becker planting? I believe some of these plants too that have been produced are also going to St. Paul. So yeah, just tell us about these new progeny families in Minnesota.
Yeah, so in the Becker planting, we have some of these crosses also. And especially with some of the Rutgers material, which like Scott was saying is, and you were saying is more cold tolerant and EFB resistant. So we're really interested in those and having, larger families, whereas in the beginning we kind of had smaller families, which makes it can make it a little bit more difficult sometimes to do our linkage analysis. And what will be interesting with some of these dialleles that we're looking at, where we're taking our best Midwest plants and crossing them by our best Midwest plants. And what I'm really interested in looking at too is we'll be able to do with that allelism tests, especially for things like EFB resistance, where we know that these plants, both of these or all of these parental parents are resistant to EFB, but we don't know if it's because they have the same EFB resistance genes. So when we cross them together, that allows us to actually do allelism tests and determine these are the same genes or these are different genes. And if they're different genes, then we can stack them together.
So is AI going to produce us the perfect hazelnut plant next year? Obviously
Not
not.
next year.
Yeah, but how do you see AI being used in the breeding program going forward? Or is it already sort of being used with these prediction models? So yeah. And it may have nothing to do with plant breeding. I don't know, but everybody knows about AI and thinks it's going to solve all the world's problems. So why not solve the hazelnut problem? So tell us about AI and hazelnut breeding.
Yeah. Well, particular problems that computers are really good at solving or can be extremely helpful in speeding things up. And so just to give an example, like I mentioned that I'm also working on chestnuts. With chestnuts, one of our current research projects is to fly drones over the trees. And because all of the burrs are produced on terminal buds in chestnut, i.e. like the crop is not in the inside of the tree crown, it's on the outside, you can see it from above. So you can get these photos of hundreds or thousands of burrs in a mature chestnut canopy, and you can train a computer using AI methods to detect what is a burr and then count them all for you. And that's great because it otherwise is, like mind-numbingly exhausting to either pick up nuts, over a 2-week period from an individual chestnut tree as they drop, or stare at a computer screen clicking on little green circles for, years on end. So that's really great. And I think that highlights one way that you can use AI is that it's really, there are like really easy to use and generally accessible and accurate computer vision detection methods. That is a field where I'd say like, yeah, artificial intelligence has done pretty well. Um, and it's not something where you're asking a computer to like measure precisely something. It's just like a counting task. That's another kind of like critical thing that I think computers might not be like great at the achieving the level of accuracy that we often need in a phenotyping method or something. Yeah, there's a lot of hype around AI. I think a lot of what we've described probably will stay the same, with maybe some tweaks around the edges, particular methods that get a little bit more efficient or change a little bit. But yeah, I'm I'm a I'm a little cautious about, some of the like the newest cutting edge stuff, including CRISPR, I should say, and that's a technique that is definitely revolutionizing functional genetics and plant breeding in a lot of crops. But in hazelnuts, in a lot of specialty crops where we don't have like single gene traits that are extraordinarily well characterized, the utility, even if we wanted to do it or had the like regeneration protocols to do it, is just a lot more reduced. So
Yeah, I mean, we really
yeah,
know almost nothing about the genetics of hazelnuts, right? In terms of single genes and their functions and
very little. Yeah, especially compared to other crops,
yeah.
or other plant and animal species. But yeah, I don't know. Emily, are you about to launch a whole AI breeding program up there?
Uh, well, my experience in my PhD with genomic selection was actually that a lot of our kind of our RR-BLUP, our G-BLUP genomic prediction models performed just as well as the more complicated ones, and they were much easier to run. They required a lot less computation power, as in like how much computation resources were required. And so I'm still a strong supporter of kind of use the simplest model that you can that's doing a good job and don't overcomplicate it if you don't have to. But AI is really rapidly evolving. And so I wouldn't be surprised if, in the next year or two, we do have really superior AI genomic prediction models that we can use. But it would also be how easy are they to run? Are they going to run in time for us to make selections with? Or is it something that's going to be computationally in unreasonable to use? So I'm open-minded about it. But I guess where I see more utility for AI are the models, the phenotyping models that you were talking about, Scott, where you're able to do object detection, like with the chestnuts, especially being terminally bearing, so you can actually see the chestnuts and you're not having occlusion of the nut itself. That helps. But my previous lab, Amanda Holzkemp's group in North Carolina, we were doing a lot with AI for object detection and working on improving when the fruit or the nut that you're trying to see is occluded by like leaves, like if the nut's behind a leaf, can the model still detect that there is a nut there? Because it can only see part of the nut. And we were able to make some really good strides in that arena. So I'm optimistic about that. And I think that would, especially if we could train models to really quickly give us at least an estimate of yield, since that is something right now we're not able often to phenotype completely because it is so labor intensive. If we could just get an estimate of how much yield is there based on an object detection from an image, that would be very powerful, and I would be very excited to use that. But developing those is a lot of work, a lot of manpower. Um, yeah, because I was
And
talking to, go
that
ahead,
really,
Jason.
yeah, I was going to say that the yield trait for the Midwest breeding program, I would say, is the most important trait in some ways, because take the Rutgers Oregon breeding programs that started with Avalana selections that had been made over, I don't know, 500,000 years, right? So they already started with a base population that had sufficient yields for commercial production. We're starting with a base population, I would argue, of American hazelnut, which has not had the base yield level to support commercial production. And so every variety that we develop has got to have the yields. right? And that requires all this yield phenotyping, which is a killer. Not only is it a huge labor cost, but it's so driven by environmental variables too, and things out of our control, like predation. You know, Blue Jay gets in there for 2 days before we get in there, are chipmunks, and we lose the data for a year. It's that kind of thing that makes it so hard. So image-based yield prediction seems to me like a great focus for work like or computer modeling like AI.
It is a
Yeah.
lot of work. Like Emily was saying, you like a genomic prediction model where you have to get the phenotypic data and the genotypic data with a AI object detection method. You need to give it some what's called annotated data. So for instance, with the chestnuts, you do have to sit there for longer than you would ever want to and circle chestnuts in order for a computer to
Train it.
know to train it to for it to know
Yeah.
what you're talking about. So that is there are enormous amounts of investment that have to go into training these bespoke object detection models. Nobody is building the tech companies aren't building chestnut detection algorithms that are just ready to go off the shelf. So yeah, it is a lot of work to build them, but if anybody has good ideas for how to use AI, there's lots of money for using
Yeah,
AI.
no doubt.
So write in and tell us what we should do.
Yeah. Well, in one of the what my previous lab, one of the things that they found that I thought was really promising, so they were doing so you can do object detection and train the model not only with images of like chestnuts, but also with just a large annotated data set of anything. And so there's these data sets online, I think one's called Coco, and it's literally like chairs are annotated. There's a picture and they're like, that's a chair, that's an object. And that information of just figuring out that there's an object there and circling it can help improve the prediction accuracies for specific things like a chestnut, just because the model is able to see that there is some kind of object there. It
Hmm.
doesn't care, but and then you tell it this is a specific object. And that supposedly can really improve prediction accuracy and decrease man hours of needing to annotate.
So in 5 years, when the agroecologist comes to you and says, look, based on markets, based on harvest, based on all these other agronomic factors, I need a plant that produces a kernel that's 1 gram in size. I need it to be 50% kernel. I need it to be no more than 10 feet tall at maturity. And I need the base of the plant that doesn't sucker very much, so it only gets maybe a foot wide. And I'd tell you that we don't yet have that plant. And do you think you'll be able to say, well, hold on, let me get into my computer and I'm going to find you two parents that if you cross them, they
Yeah.
are likely to produce that plant that you're looking for. Will you be
Yeah,
able to do that
exactly.
for me?
Right. I mean, I know that that's been the ask for years, but I do think that in 5 years that would be definitely an achievable goal to not just be able to say this is the quote unquote best plant, but that can be defined any number of ways. And so what the predictive tools allow you to do is not just, sort a list of plants from highest yielding to lowest yielding, but really to make the prediction, what is the yield or what is this base width of the plant, et cetera, specifically. And so as breeding targets change or evolve or get refined, those predictions can immediately be used to find the required parents or screen through large seedling families. So yeah, that would be one thing that I would really hope we get to. Another thing I would hope we get to is just a more refined phenotyping a set of phenotyping protocols. One of the challenging things with breeding a crop like hazelnuts, where it is still being there's a ton of research, maybe more important research going into how these plants should be grown at scale, how they need to be managed, how they should be harvested, marketed, handled after harvest. So just to take harvest as the example, how we want these plants to look and grow, and it is going to be determined by what kind of harvesting methods we have, and what kind of rows, plant spacing we want, and we're still figuring those things out as we go. And that's kind of, Emily referred to this as also just like almost like just a domestication event that we are part of here, as well as sort of a traditional plant breeding program. So I hope in 5 years that a lot of these agronomic and cultural management questions are sufficiently resolved that we will have figured out exactly how we want to measure traits and do it efficiently so that we can sort of build the models that we need for all of these traits.
Okay, final question. I'm going to put you both on the spot. You got to answer immediately without hesitation to prove that you're true hazelnut aficionados. Ready?
Okay.
What's your favorite hazelnut food product that you eat all the time?
Just the hazelnuts,
Perfect.
the brined ones.
Oh, brined. Good
Yeah.
choice.
Roasted. I am allergic to raw hazelnuts.
Oh, really?
I have that betula allergy. It's not due to breeding hazelnuts my whole life, which is a common thing that happens to plant breeders, is they become allergic to their crop. But no, I just can't eat them raw, so I got to eat them roasted.
Brined and roasted, I like it. Purists, neither of you went right to the chocolate hazelnut spread, so it's good.
No.
Approved.
Brought to you by the University of Wisconsin-Madison Division of Extension.